Good evening and welcome to the Science in the News lecture series. This is our lecture titled Fat versus Sugar, the Culture of American Dieting. Thank you for joining us tonight. Throughout the lecture, please feel free to fill out the form below to give us feedback on your viewing experience, as well as to ask any questions you might have. We will ask these questions during the question breaks that the speakers will pause for during their portion of the lecture. Again, thank you for joining us tonight, and we will be getting started in just a few minutes. All right, good evening, everyone. Welcome to the Science in the News lecture series. This is our 15th year running this lecture series, so we're happy that you are all here with us tonight. This is our sixth lecture of this series called Fat versus Sugar, the Culture of American Dieting. So if you've never been with us before, Science in the News is a graduate student group at Harvard University. We encompass all of the Graduate School of Arts and Sciences, and our main goal is to provide a fun, interactive events to bring together non-scientists and scientists so that we can learn from each other and have great conversations about the current research that's going on in all areas of science. So we've been running our lecture series for 15 years now, and in addition to our fall lecture series, we also have a spring lecture series. This one occurs on the Cambridge campus, so keep a lookout for that in the spring. But in addition, we also have another fun event we want to tell you about called Science by the Pint. This event occurs the second Monday of every month over at the Burn in Davis Square, which is a local pub. And at those events, we invite a scientist and members of their lab to join us, and they give a short talk about what they do and then spend the rest of the time mingling from table to table and talking and answering questions about their research. So it's a really fun, casual event. You can speak with a scientist over food and beer, and the next one we have will be on November 10th. With Dr. Sasolov, a weather report from an exoplanet, clouds and rain in a place 40 light years away. So it should be a great one. And also, in addition to that, we always have available on our website articles for you to check out. These are called Signal to Noise. So if you didn't already, you can pick one of these up and check out our website for those. But for now, Vinny's going to tell you about what will happen tonight.
All right. Well, it's good to see a lot of familiar faces in the audience today. For those of you who are newcomers, just to give you a little brief intro to our format, each of our lectures is split into 3 parts, lasting about 30 minutes each. And today you'll hear from these lovely ladies, Katie, Abby, and Mary, and they'll be telling you about fat versus sugar and the culture of American dieting. The way each of the segments works is that we have about a couple of question breaks in the middle where you can ask the lecturers the questions. And we ask that you hold your questions up until those times, and you can also feel free to ask them more questions either via email or during the breaks that we'll have. We'll have a break between the second and third part of the lecture. If you also notice the speakers, when you ask them questions, they'll be repeating the questions, and this is because we have an audience on the live stream, so hello to everybody on the live stream. So in the event in the future, if you guys aren't able to make it in person, you can stream live and also ask questions to the lecturers. And finally, I want to bring to your attention the surveys that we have. This helps, if you could fill these out, that would be awesome, because it helps tell us what we're doing right, what we could improve on, and any topics that you guys would like to hear about in the future in our lecture series. And actually, finally, really now, at the end of the lecture, these ladies will also have a demo for you that relates to some of the concepts that they're covering today in the lecture. So without further ado, I will turn it over to Katie.
All right, hello, and thank you for coming. Good evening. My name is Catherine Richardson, and I will be opening for tonight. We'll be talking about fat versus sugar, the culture of American dieting. So I will be your waiter or waitress for the first part, and your meal for this evening will consist of 3 courses. The first being the building blocks or macronutrients that you need in your diet. The second will be, how do scientists evaluate diets? Mary will go into a low sugar or low carbohydrate diet compared to a low fat diet, and how we as scientists evaluate these types of diets. And finally, Abby will finish on how society has changed food, particularly discussing artificial sweeteners and gluten sensitivity. So as I mentioned, I will be covering the building blocks, the A, B, and C's that you guys need to understand nutrition and diets, as well as understand the rest of this lecture. So let's get started. After the first course, you'll understand what the purpose of food is. You'll understand our 3 macronutrients: carbohydrates, proteins, and fats. For each of them, you'll understand, what are they made of? Why are they important? And how does the body actually use them? And then we'll end with how much. How much of these macronutrients should we be eating in our balanced diet? So I thought we would start off with a really fun social experiment, because we all love those. We're stuck on a deserted island. You're allowed to bring an unlimited source of water and one of the food items below. Oh, sorry. Yeah, we're really deserted. No, nothing. All right, we're back to the deserted island. Okay, we're on the deserted island. You're allowed to bring an unlimited source of water and one of the food items below. You can choose from corn, hot dogs, peaches, or milk chocolate. So I want to see you guys raise your hand for which food you would choose. So 1 for corn and 4 for milk chocolate, etc. I've seen a lot of people choosing corn over here. I think that's what I would choose if I was just deciding on what I wanted. It's a good array. I see some hot dogs, some peaches. So the purpose of this exercise was to really bring to light what we think of food and what food is used for. So if we were thinking about what food we would want to bring based on the best nutrients and how long we would live, the correct answer would actually be a hot dog, because hot dogs contain all of the fats, proteins, and carbohydrates that we need to live. But eat hot dogs every day for the rest of our lives. And this is where the psychological aspect of food comes into play. We eat what we want to eat, not what our bodies actually need. Which brings us to the purpose of food. It's not to make us feel better, even though we want it to. It's not to eat when we're bored, and it's not meant to prevent the growlings in our stomach. Food is actually used to give our bodies energy, and solely energy. The energy that we get from food is used for basic survival, which includes organ function, it includes maintaining our body temperature, and it includes blood circulation as well as breathing. The energy that we get from food is also used for us to do work, for us to think, move, exercise, anything external. Now, if we eat calories, calories are the unit by which we measure food. And they're not tiny creatures that live in our closets and purposely sew our clothes tighter every night so we just can't fit into them the next day. Calories are a unit of energy. In fact, 1 calorie is the amount of energy needed to raise the temperature of 1 kilogram of water by 1 degree Celsius. And if you're curious about how much energy or calories your food contains, you can find them here on nutrition labels that are on the back of your food. Now, to put this in a more practical grounding, 1 calorie is equal to the amount of energy it requires for your heart to beat about 4,000 times, or for the average person to walk about 20 feet. When we eat excess calories or excess energies, let's say we go to JP Licks and we get that extra large sundae, when we use up that energy to survive and to do our work, we have energy that is left over, excess energy, that can't be destroyed. So what happens? We store it as the dreaded fat. But it's not all bad news. If we eat fewer calories, we won't have enough energy to use all of our survival and do all of our work. So we'll take some of that dreaded fat and we'll break it down and use the energy from that fat to fuel our organ functions and any extra movements that we're doing. Calories, these energies, come from 3 main sources of macronutrients. Those include carbohydrates, proteins, and fats. Information about each of these macronutrients can be found on your nutrition labels. And the purpose of these macronutrients are to give your body energy. We're going to go through each of these macronutrients and understand what they're made of, why they're important, and how our bodies use them. So let's start off with carbohydrates. Carbohydrates are made of simple sugars. That is correct. Carbohydrates are sugars, and sugars are carbohydrates. There are 3 main simple sugars: glucose, fructose, and galactose. These 3 simple sugars can combine in pairs of 2 to form table sugar, or sucrose, or milk sugar, which we also call lactose. These pairs of simple sugars are what we normally refer to as sugar. These simple sugars can also combine in multiples to form what we call complex carbohydrates. You can see starch is an example where there are multiple glucose subunits. Fiber is also an example of a complex carbohydrate. We can't talk about carbohydrates without touching upon one of them that's in the news quite frequently, which is high fructose corn syrup. High fructose corn syrup, believe it or not, comes from corn. Corn is largely made of a starch, which we discussed before. Through a chemical and artificial process, this starch is broken down into its individual units. And some of those individual units are converted from the glucose into a fructose. And this is what constitutes high fructose corn syrup. The composition is surprising. It's only 55% fructose compared to 45% glucose. If you compare this to a very common sugar or table sugar, when table sugar is broken down, it's 50% fructose and 50% glucose. So the difference between high fructose corn syrup is a 5% more fructose. Where do we find high fructose corn syrup? We find it in soft drinks, salad dressings, ketchups, breads, cereals, yogurt. It's in a lot of food items. So why do we use it? We use it because it's cheaper. Boils down to that. It's also sweeter. Fructose is sweeter than glucose. It's easier to transport because it's a liquid, and it has a longer shelf life for food. But I'm sure many of you have seen that high fructose corn syrup has been linked to a variety of health risks. These include fatty liver, or fat deposits in your liver that prevent liver functioning. It also includes bad cholesterol, heart disease, and it's linked to obesity. It's important to note that these health risks are largely seen when there is an excess of high fructose corn syrup that's being consumed. But it's really easy to do so since it's in all of our foods, or almost all of our foods. So to understand how these simple sugars, whether they're glucose, fructose, or galactose, are used by our body, we need to understand the digestive system. So just a basic review. Our digestive system comprises our mouth, esophagus, stomach, our small intestine, and our large intestine. Sugar, starches, and dietary fiber are ingested through our mouth and move through our stomach. When sugars and starches reach our small intestine, they're broken down into their individual simple sugar units. These individual simple sugar units can then be absorbed through the walls of our small intestine and enter our bloodstream, where our cells can then get access to them. Dietary fiber is a little different. Dietary fiber moves straight through us, all the way through our digestive system. That's because our body lacks the ability to break apart these bonds or connections or links between the glucose simple sugars in dietary fiber. That means that our body isn't absorbing any of that dietary fiber, so we're not getting any energy from it. So technically, dietary fiber doesn't have any calories in our body. Once glucose or any of the simple sugars reach a cell that needs those simple sugars, for instance, a muscle cell, the glucose cannot readily enter the cell. In fact, we need another hormone known as insulin. Insulin is triggered in response to glucose levels in your blood. And that insulin travels in your bloodstream and allows the glucose to then enter your cells. Once the glucose is in your cells, that glucose can be broken down through a series of chemical reactions to generate energy so that we can use our muscles or use our brains or whatever cell needs energy. Of energy for most of our cells. But it's important to talk about how our body deals with fructose. So as we mentioned, fructose is in high fructose corn syrup, but it's also in table sugar. And fructose really only enters our liver cells. And it does not induce insulin production, nor does it require insulin to enter our cells. However, once the fructose is in our liver cells, we largely don't use it for energy. We convert it to fat. This is why high contents of high fructose corn syrup with fructose in it can lead to fatty liver syndrome, because when we ingest too much, that fructose is put into our liver and stored as fat. At this point, I want to do a review of our carbohydrates before we enter our first questions period. So carbohydrates are made of simple sugars. They're absorbed in our small intestine. Glucose is the main source of energy for our cells, and it requires insulin to get into our cells. Any excess glucose that we don't use for energy is stored as fat, even though it's a carbohydrate. And finally, fructose is largely stored in the liver as fat. So now that we're done with carbohydrates, I want to break for questions. Yes, of course. So the question is, why is everyone freaking out about high fructose corn syrup when it only contains a little bit more fructose than regular sugar? Which is a great question. One of the reasons is that our body can access it better. So high fructose corn syrup is already broken down into its glucose and fructose in the liquid form. So it's easier for our body to absorb that fructose. Another reason is that when you're eating foods with high fructose corn syrup, it's really easy to eat too much of them, more so than with sugar, because it's sweeter and it's used in almost anything. So when we're ingesting those high amounts of high fructose corn syrup, that's when we lead to problems. But if we were ingesting similarly high amounts of sugar, we would also have some of these problems. It's just the accessibility to the fructose would be different. Up there. Right, you in the brown sweater, yes. So fructose will always be stored as fat in the liver, but it gets back to the same idea of accessibility. So the fructose that you might find in fruit can be bound up to other things that are in the fruit and can be less accessible to be absorbed in our small intestine. Exactly. It can still get into the cells, but I don't want to tell you guys don't eat fruit. There are lots of benefits of fruit. You should be eating fruit, even though it might have some fructose in it. It's just the amount of fructose that you're eating on a daily basis. Right here. So the question is, do some people absorb fructose more or less than other people? Which is a great question, and it gets to the variability that exists in our population. I'm not aware of any studies that have actually measured how available fructose is between individuals, but that is certainly possible. And the reason for our bodies handling fructose differently is that we could absorb it differently, more or less between people. One more. So the question was, is there any benefit to having the fat in the liver? So the liver is a spot where we store fat, sometimes as an intermediate before we bring it to adipose tissue or our fat cells. There is no large benefit to storing excess fat that we get from high fructose corn syrup in our liver. So because we're ingesting fats too that are also going to be stored as fat, we're just overloading the amount of fat in the liver, which can overload the liver cells and they can start to die. So generally, there is a role of some fat, but we're getting it from our regular fat to begin with, not just from our high fructose. So I'm going to move on to the second part, but there'll be more time for questions after the second part and during the break, okay? All right, so we've done carbohydrates. So let's move on to proteins really quickly. Proteins are made of amino acids. There are 20 unique amino acids, and they're listed here. These amino acids combine in various combinations to form longer chains, which we call proteins. Proteins are actually essential in our diet. So we can't eat all carbs, regardless of how much we want to. We need protein. And that's because 9 of these amino acids are essential, meaning that our body does not make them and we're required to have them in our diet. Proteins are digested similarly to sugars and starches, but there's one main exception, and that is when the proteins move through our stomach, they're partially broken down. Proteins require more energy and are harder to break apart than carbohydrates, so it starts in the stomach. Once you reach the small intestine, the proteins are completely broken down into their individual amino acids. Once they're broken down into their individual amino acids, they can cross the small intestine, move into our bloodstream, and our cells can have access to them. So what do our body do with proteins? Well, one of them that I'm sure all of you can think of is build and repair those muscles. So those proteins are used to make new proteins in our body. But they're also used for a variety of other purposes, for bone health and bone reforming. They're also used in our hair and our nails. They're mostly made of protein. And finally, they're extremely important in proper immune function and scarring or healing of tissues. Proteins can be used as energy, but only in circumstances where our other macronutrients, our carbohydrates and our fats, are in very low quantities, where we need the energy from the protein. So let's do a basic review of proteins before we get on to our fat section. Proteins are made of amino acids. There are 20 unique amino acids. They're absorbed in the small intestine, and they're essential in our diet. In fact, 9 of the amino acids we cannot make, but we must eat. Amino acids are used in the body to make new proteins like our muscles. They can also be used as energy, but yet again, too much of those proteins can be stored as fat, even though they're proteins. So let's move on to our third macronutrient, which is fat. Fats are made up of fatty acids. There are 2 main types of fatty acids. saturated fatty acids, and unsaturated fatty acids. The difference between these fatty acids is their structure and how they're held together. These fatty acids combine in threes to form fat or triglycerides. Here's an example of a fat which has 3 saturated fatty acids which combine to form one fat. The fatty acids can also combine to be saturated or unsaturated in one fat molecule. So we're going to move on to our final game, which is guess that fatty acid. So we have group A and group B. Group A is solid at room temperature. It's considered a bad fatty acid, meaning it's associated with heart disease. And it's also increasing our bad or lousy cholesterol. An example of group A fatty acid is butter. Group B is liquid at room temperature. It's considered to be a good fat, and it increases our good cholesterol. And this is called, an example of this is olive oil or vegetable oil. So I want to see a raise of hands if you think group A is saturated fatty acids. The majority has it. You're correct. Group A is saturated fatty acids. They're considered to be bad fats, whereas the unsaturated fatty acids found in oils are considered to be good fats. Let's move on to another hot topic, trans fats or trans fatty acids. Trans fats are actually made from unsaturated fatty acids, such as vegetable oil or olive oil. What happens is there's a chemical and artificial process called partial hydrogenation, where people add hydrogen atoms to our unsaturated fat. This causes the unsaturated fat to change shape and form what we know as trans fats or trans fatty acids. These are also called partially hydrogenated vegetable oils. So if you're ever curious if your food contains one of these, you can look at the trans fat on the nutrition labels or just look at the ingredients for a partially hydrogenated oil, such as vegetable oil. Vegetable oil contains a lot of trans fats. What do we find trans fats in? Any of that really good fried food, if it's fried in oil, that contains trans fats. We also find it in frosting, and non-dairy coffee creamer can have a lot of trans fats in it, so always look at your labels. Why do we use them? Again, it's cheaper, they're easy to transport, and they have a long shelf life. But they're associated with some health risks, including heart disease, diabetes, and high levels of bad cholesterol. Now you may be thinking that fat is just really bad for us, but that's not the case. We actually need fat in our diet. There are 2 fatty acids that are essential fatty acids, meaning that we must consume them in foods. These are omega-3 and omega-6 fatty acids. You might be familiar with omega-3s from fish oil or really oily fishes. They contain a lot of omega-3s. And these fatty acids are essential. They're required for our blood to clot. They're required for our immune system to function normally. And they're also required for brain development and brain functioning. So all of that thinking you guys are doing right now, you need these omega-3 and omega-6s. Fats are digested similarly to proteins and carbohydrates. They move through our mouth and into our stomach. And once they reach the small intestine, they're broken down into our fatty acids. But there's a unique part of the absorption through the small intestine. When fatty acids move across the small intestine, they reform to be a fat. But this fat cannot travel in our bloodstream and get to our cells easily. We need to coat this fat in what we call a lipoprotein, which for the rest of this lecture will look like a suitcase. So these lipoproteins carry around fat and cholesterol in our bloodstream because normally they wouldn't be able to move. So once they're coated in their lipoprotein suitcase, they can enter the bloodstream. Once in the bloodstream, they can reach a cell that might need energy or fat. Then those fatty acids can release from the suitcase, move into the cell, and be used as energy or again, stored as fat. You might know a lot about these lipoproteins and how they associate with cholesterol. So I want to touch on cholesterol. Cholesterol is actually a type of fat, and it's not bad. We need it. It's essential. It coats our cells and helps our cells maintain their structure and their function. It's needed to produce those ever so important estrogen and testosterone hormones. And it's carried and needed for our lipoproteins, which carry our fats and additional cholesterol. There are two types of these lipoprotein suitcases that we hear about commonly. One of them is called LDL, or low density lipoprotein. These are considered to be lousy or bad lipoproteins. And the other is HDL, or high density lipoprotein. These are considered to be healthy or good lipoproteins. The difference between these two lipoproteins is in their function. So LDL, or our bad lipoproteins, carry our fats and our cholesterol from organs such as the liver into our bloodstream so that our cells can get access to them. But when we have too much of our LDL or too much of our bad cholesterol, it can get sticky and group together in our bloodstream or in our arteries. When this happens, they form what we call plaques, or groups of this LDL that clump together and stick to the side of our arteries. This impedes blood flow through the artery and is associated with heart disease. HDL, or the good lipoproteins, can actually break apart that plaque and bring some of the fats and cholesterol back to the liver to then be degraded and broken down and excreted. So the difference between the two is where they're moving fats. So the HDL can break apart those bad plaques and move them back to the liver to be excreted. So let's do a review of our fats. Fats are made of fatty acids. There are two types, saturated and unsaturated fatty acids. Trans fats arise from unsaturated fatty acids that undergo a process called partial hydrogenation, which turns them into trans fats. Not all fat is bad for you. We need those omega-3s and omega-6 fats. Our lipoprotein suitcases package and carry fat and cholesterol through our bloodstream to get to our cells. There are two types, LDL, or the bad lipoproteins, which move the fat and cholesterol away from our liver and into our bloodstream where they can form plaques. And finally, HDL, or good lipoproteins, that can move fat and cholesterol out of our arteries and back to our liver to be broken down. Now that we understand our three macronutrients and what they are and what they're made of and how our body uses them, the grand old question is, how much of these do we need to eat every day? So the FDA suggests a diet of 2,000 calories a day. This may vary, and it does vary, based on a variety of factors. So it's important to consult your physician to find the best calorie amount that you should be consuming in a day. But in the FDA suggested diet, they suggest 2,000 calories made up of 320 grams of carbohydrates, 64 of fat, and 55 of protein. Information about how many grams you're ingesting can be found on your nutrition labels. And importantly, your percent daily value suggests how much until you reach the 100% of your allotted grams. So for instance, the 12 grams of fat found in this macaroni and cheese is 18% of the 64 grams suggested to consume. However, it gets a little tricky because you need to look at the serving size. So this serving size is just for one serving, but in this container of macaroni and cheese, there are in fact two. So if you eat the whole container of macaroni and cheese, you'll be ingesting 24 grams of fat. So it's important to be looking out for these differences in labels and serving size. When we look at what the average American consumed in 2010, it's staggering. The average American consumes 3,770 calories a day. And of that, the amount of macronutrients are drastically higher than what's suggested. Overall, you can tell Americans are overeating. So with that, I'd like to take home points. The first being that food is fuel to provide our bodies with energy. That energy comes from calories, which we find in our 3 macronutrients: carbohydrates, proteins, and fats. And any excess calories are converted to that dreaded fat and stored as fat. Thankfully, we are not on a deserted island, and we do not have to eat that hot dog every day. But it's important to maintain a balanced diet of macronutrients and make sure that according to your physician or your lifestyle, that you're eating a balanced diet of proteins and carbohydrates and fats. So with that, I'd like to take any more questions before we move on to our next part. Yeah, you had a question for us. So the question pertains to sugars and carbohydrates. This audience member points out that vegetables do contain carbohydrates. So she's asking how that pertains to what I've been talking about. So vegetables are a source that are particularly high in dietary fiber. And so that dietary fiber, as we mentioned before, is not considered to be a caloric macronutrient because it's not absorbed. Vegetables also have a lot of really other good things in them, vitamins and minerals that we need. And the carbohydrates in them are significantly less compared to what we think of as breads and potatoes and pasta. So it's important. You need carbohydrates. It's just how much and figuring out how that fits in with your diet. You can eat a ton of vegetables, yeah, like your mother always told you. Any other questions? Right here. Yes. So the question was, how do these macronutrients relate to how we taste them? There seems to be a correlation. What tastes good is bad for us. So how we taste things are based on 3 simple receptors that are on our taste buds. The way we taste sweet things is through a receptor called the TR2 receptor. Sweet things bind to this receptor and tell our brain this is sweet. Fructose, for example, stays on there longer and sends those good signals that this is sweet to our brain for a longer period of time. And those communications of what's good and what's bad can vary based on people. But it is true that many of us taste sugar and we think, that's really, really good. And that's due in part to the receptor and how it communicates to our brain. Any other questions? I thought I saw a hand over here. Yeah. Okay. Mm-hmm. So the first question was, I mentioned that excess proteins can be turned into fat, but if you're eating under your caloric value for the day, will those proteins be converted to fat? And the answer is no. So you need a lot of excess proteins for that to go into fat. Yes, right here. The question was, is there a debate in our scientific community over that pie chart that showed the amount of macronutrients we should be eating? And the answer is certainly yes. There's a huge debate in it. This is just what the FDA suggests. It can vary for a variety of reasons. One of them being your age, one of them being your gender, one of them being your activity level, one of them just being that we're all unique human beings. And Mary's going to talk about how these macronutrients are used in our body and how that can be a little confusing based on variation between people. Yes, over here. Yes.
So is LDL actually essential? Or should we, how does that work?
So the question was, we say LDL is bad, but is it really essential or is it really bad? So the answer is we do need LDL. It's not bad. We don't want to get rid of it altogether. It's what brings that cholesterol and fat to our cells when we need them. So if we get rid of it's just maintaining how much of it we have in our bloodstream. Yes, right there. Mm-hmm. Mm-hmm. So the question was about MSG, which we often find in Chinese food, and how that compares to simple amino acid that it is a sister to or is like. So MSG, a lot of the studies that suggest that it's bad are correlative studies. So they say, oh, look, you're taking MSG, you're having a bad reaction to it. And so the science behind that, in my knowledge, is not completely known of what is actually so bad about it, but it does have a different structure and can be used differently in our bodies. So I think we have time for more questions. So I'll take one right here. So the question was portion sizes on the label. Should we trust them? Which is a really interesting question. You know, I never really thought of it. I just take those nutrition labels to heart. I haven't tested it, whether or not we should believe them or we shouldn't believe them. We certainly can with a scale at home, or part of our demonstration today is actually going to be using what's known, which is used to measure how many calories are in one food source. So you can test that as well. If you do test it, I would be very interested to know the results. So I think with that, if there are other questions during the break, feel free to come up and ask me. Otherwise, as I mentioned, the demonstration at the end will show you how we actually determine how many calories are in one food source. So make sure to stick around if you're interested. And I will turn it over to Mary. Thank you. Oh, Mary, you make this. Okay, great. Turn this on. And then, okay. So hi, my name is Mary Gearing. And now that Katie has given us such a great background, a great foundation and understanding of macronutrients, I'm really excited to lead us through how scientists evaluate diet. And one of the reasons why I'm so excited about this topic is that nutrition gets a lot of press. So the articles I'll be showing you here all came from the New York Times in the past year. And these articles tend to fall into two categories. The first being why nutrition is important. So we see a lot of articles about how Americans are getting fatter, our obesity epidemic, beating back the risk of diabetes, how proper nutrition can lower our rates of chronic disease. And then we also see new findings in the field, new things that are complicating what we know about nutrition. So examples here, new answers about carbs and fat. We'll be coming back to this throughout the lecture. These advice columns, advice from a vegan cardiologist. Is breakfast overrated? So turning what we all have thought to be true and what we've all been told just on its head. And my personal favorite article, why nutrition is so confusing. And so I hope after the end of this section that we'll be able to demystify a lot of this and really look beyond the headlines and see what studies are really asking and what their conclusions are really saying to us. So your overview for this second course, I'll go into why we need human studies. Katie told us all about macronutrients and how they're digested. We know all of that. Why do we still need human studies? And then we'll just dip our toes into research with a short-term and a long-term case study. So why do we need human studies to evaluate diet? First of all, what is a diet?
So most of us are familiar with this definition of kind of going on a diet, eating less in order to try to lose weight and be healthier. But what we may not realize is that a diet is also just what we eat normally. If I asked all of you to take out a piece of paper and write down what you've been eating for the past week, that would be your diet. So the point here is that we're and our diets influence our risk of chronic diseases like heart disease and diabetes. And so it's really important that we get these right, that we know what sort of diets are healthiest for us. And so here, just to review Katie's presentation, calories come from our 3 macronutrients: carbohydrates, proteins, and fats. And again, we understand these pathways really well. We understand how these macronutrients are metabolized, how they're broken down and utilized. And so here, just showing glucose entering a cell with the help of insulin, and then showing fats being broken down, packaged into our favorite suitcases, and then sent into the bloodstream. But the problem is that the overall picture is a little bit more complicated than that. With almost every food we're eating, and of course with our whole diet every day, we're getting all of these things: fat, carbohydrates, and protein in different amounts. And what we really want to understand is how eating macronutrients is going to affect our health. So how person A on the left here, who's eating lots of fruits and vegetables and unprocessed protein sources like eggs and fish, compares to our more typical American diet of meat and refined carbohydrates. And so for this, we need our human studies, and these studies really come in all shapes and sizes. So if we look based on a timeline, we can see maybe a short 1-year weight loss study. A study gives us some time for follow-up, and some studies may extend as long as 20 years. We also see differences in the number of subjects. So here we can imagine this one stick figure standing for 100 or 200 people. We may have a few thousand, or even one of the studies I'll talk about today, 100,000 participants. But no matter what the differences in these studies, they can all be reduced to fundamental questions. So I want you to think back to elementary school where you learned about the W's. And so in my case studies, I'll be referring to these words constantly to really take you through the studies and figure out what they're all about. So we'll start with our short-term case study. And this one is very new to the news. It came out of Tulane University earlier this year. So being scientists, we always want to start with a why. We need a rationale for what we're doing. And so here our rationale is twofold. So the first right here, this organ may look familiar: heart. Heart disease is one of the leading causes of death in Americans. And nutrition, as I said earlier, can play a role in its prevention. The other here is our obesity epidemic. So this is a map that's colored based on the rates of obesity in these states in 2011. So the darker orange indicates obesity rates of over 30%. Massachusetts is doing okay, but still nothing to brag about at about 20%. And where they did this study, Tulane University in Louisiana, again, is at about 30%. So who are they recruiting here? So they're recruiting obese participants. They have about 150 of them. And these are people who don't yet have a chronic heart disease, and they don't have diabetes. And so what are we looking at? So here we're comparing a low-carb and a low-fat diet. We want to define this a little bit more clearly. So our low-carb diet, they're eating about 30% calories from fat. Sorry, this is actually supposed to be low fat. I'm not sure how that got switched. So here it's the amount of fat that you would get from about 3 to 4 hot dogs if we go back to Katie's original deserted island example. Whereas our low carb diet is limited to 40 grams of digestible carbs. So this is our total carbs minus fiber. And this ends up being about 2 slices of bread. And then when are they measuring this? This is a short-term study. And so they're going to look at these participants at 3, 6, and 12 months. So what you may be saying looking at the slide right now is, this low-fat diet doesn't seem too bad. You know, I could probably limit my fat to 3 to 4 hot dogs worth. That doesn't seem too hard. The low-carb diet seems a little bit more difficult. Limiting to the amount of carbs in 2 slices of bread That's a pretty low carb diet. And we can see this even more clearly when we look in comparison to what the FDA recommends. So here, just our familiar pie chart for our FDA diet. We see a very high percentage of carbs and then a substantial amount of fat and protein. So the low fat group, they were fairly similar to this, a little less carbohydrate, a little more protein. But again, we see very drastically this difference in the low carb group, that they're here replacing, they are increasing their protein as well, but much of the increase is coming from fat. So here we're just making the point that these people are eating the same number of calories, it's just that their distribution is different. And so how are we going to look at these diets? How are we going to see if the low carb or the low fat is more healthy? So the first thing we want to look at is weight loss. So we all love the feeling of, you know, dropping a pant size or a dress size, losing numbers on the scale. But it's important to realize that weight loss is not the only indicator of health. So you can imagine if you went to that deserted island and you didn't have any hot dogs and you lost 20 pounds, you're not increasing your health even though you're losing weight. So we have to look at a couple of other things. So the first thing we want to look at is fat mass, so the percentage of your body that's made of fat versus lean mass. So this right here is a muscle. You may have heard that muscle weighs more than fat. Not exactly true. It's just that muscle takes up less space, which is why if you have more muscle, you tend to look thinner and slimmer. Again, we're going to look at our cholesterol, so our HDL, our good healthy cholesterol, or our LDL, our lousy cholesterol. And again, just our familiar suitcases. So just reminding you that LDL, if we have too much of it in the blood, can then stick to these arteries, create plaques, and this impedes blood flow, and this will result in heart disease. Whereas our HDL has a protective effect. It's able to remove cholesterol from these plaques, return it to the liver where it can be converted. Okay, so let's go ahead and get to the results. And so I'm just going to show you a few charts from this paper. So first of all, we're looking at our change in body weight. And here we're looking in pounds. So 0 pounds, our starting weight, 5 pounds lost, 10, 15, and 20 over a period of 12 months. So if we first look at our low fat group, we see they lost about 5 pounds, whereas our low carb group had a much more impressive weight loss. They lost closer to 10 pounds and kept it off. Now if we look at our fat mass, like I told you earlier, so we're looking at it as the percentage. And here, 0 our starting, and then losing 1%, 2%, 3%. We don't really see a change in fat mass in the low fat group, but in the low carb group, we see a consistent about 1% decrease in fat mass. So one thing that I didn't mention are these little lines here, these vertical lines. And basically what these are doing are showing that there are differences in the people that were given these diets. So if you just took Abby, me, and Katie as an example, if you put us all on a diet, would you expect us to all react exactly the same way? No. Common sense, right? So one of the things we have to take into account in human studies is variation, how we're all going to respond naturally a little bit differently to the same treatment. So I just want, if anyone wants to yell out, if there are any basic characteristics you can think of that would affect weight loss. Just go ahead and yell them out. Exercise, age. Yep. Gut microbiome. Yeah. So we have everything from very kind of scientific things, our gut microbiome, our metabolism, our genetics, to more obvious things like age and gender. And so what I want to get to as a point here is that in these studies, you need to control for these things. So when they're determining groups for the low fat and the low carb diet, these are just a few of the things that they had to evaluate to make sure they were the same between the two groups. So we already hit on age and then gender, the percentage, they measure this as the percentage of people in each group who were female. And so if we look at just the numbers, we can see they're about the same for both groups. And this is good because we want to know that we're not seeing a difference in the two groups
because, say, one of them is a lot younger or one of them is composed of mostly men versus mostly women. So just to have an overview of the study. So as I mentioned, we saw decreases in body weight for both of the groups, but we saw greater decreases in the low-carb group. And then the low-carb group was the only group to show an increase in lean mass, so more muscle, and then a decrease in fat. And we didn't see any decreases in our bad cholesterol, but we did see increased good cholesterol in both groups, again with the low-carb group having a greater increase. So the strengths of this study. So first of all, it's comparing two groups directly. And so this study design allows us to talk about the direct effects of diet. To say this low-carb diet caused greater weight loss. Close monitoring and consistency. So this is really important. When you do a study and you tell someone, okay, now eat a low-carb diet, you want to make sure they're actually eating a low-carb diet. Otherwise, that's really going to change your results. And so since this study was done over such a short period of time at one university, they were able to keep very close tabs on their participants. The other thing is related to monitoring high compliance. So through dietary studies, they were able to establish that these participants were eating appropriately. So weaknesses of this study. Sometimes it's almost more important to look at the weaknesses compared to the strengths. And I think for this, we really have to go back to our fundamental question, why? What was our motivation for doing this study? And so remember I said it was twofold. So I talked about heart disease and the obesity epidemic. And so the problem here is basically what we can't see because of this short time frame. So if we look at what we can't see, the first is possible weight regain. I'm sure we all know a yo-yo dieter who will lose 30 pounds, then gain 20 back, and then maybe lose another 50. So 1 year doesn't allow us to look at this phenomenon. We're really just looking at short-term weight loss. The other is changes in disease frequency. 1 year is not a long enough time to allow for people to develop heart disease. So what we're looking at actually is what we call surrogate outcomes. We're looking at LDL and HDL and other blood parameters to try to predict the risk of heart disease, but we can't actually look at heart disease itself. And with that, I'll take a few questions. Yes. Yes. So correlative are human studies with experimental studies in mice. So this is a really important question because it's a lot more feasible to do studies in mice. There are fewer regulations, and it's also just mice are standardized. They tend to be inbred, and so you don't see as much variation as what I showed you here. The problem is that mouse metabolism is very different than human metabolism. So my lab actually studies cholesterol and heart disease. And compared to mice, humans are much more susceptible to heart disease. Mice have much lower levels of bad cholesterol and much higher levels of good cholesterol. And so this, we say that this kind of confounds our results. So there haven't been, to further answer Jake's question, there haven't been a ton of studies that compare low carb and low fat diets in mice. There was a very large study recently that looked kind of across the board at diets with varying carb, protein, and fat intake. But again, the differences in mouse metabolism and human metabolism, and also the way mice are housed, they're basically given as much food as they will eat. It's just placed in their cage and they can eat at will, does reduce the value of mouse studies especially for diet. Yeah, so the question is there another animal model that would be better than mice? So there's always something better. So basically what we want is something that's more genetically related to humans. So actually mini pigs, so very small pigs, are much more closely related to humans, especially in terms of cholesterol metabolism. The problem is, again, it's going to cost much more to house a mini pig than to house a mouse. But also, in addition to mini pigs, you can use rats, you can use monkeys as well, but again, it's even more prohibitively expensive. Yeah. Yes, so Steph's question is all about adherence. So how do they adjust for the fact that people may not adhere to these diets? So this is all through dietary recall. So they were asked to write down on a sheet what they had been eating over the past 24 hours. And this was used to judge how well they were adhering to their particular diet. I will mention another thing from this study that hopefully boosted adherence is dietary counseling. These participants were subject to dietary counseling weekly at the start of the study and then bi-weekly later on. And this by itself has been shown to increase adherence and also increase weight loss. Okay, and with that, we'll move on to our second case study. So here we're looking at a long-term case study. And again, our first case, and this is actually going to be a 20-year study. So in picking this long-term case study, what I wanted to look at is really the relationship between long and short-term case studies. So after our short-term case study, you may be thinking, okay, low carb is great. I'll go home, I'll throw out my bread and my pasta, and I will restrict my carbs. The problem is that it's not that simple. As you can tell from the title here, nutrition is heavily debated. So I just want to show you what happened in just one media outlet, the New York Times, in response to this study. So it was first published as a call for a low-carb diet that embraces fat. A couple days later, the opinion section posted an off-talk, a debate fueled by carbs or lack thereof, in which they consulted a number of nutrition experts to weigh in. And then a day later, a feature was posted on their health blog, the Well blog, new answers about carbs and fat. And the funny thing about this is that a low-carb diet is not exactly a new idea. The diet that these people were following is actually a modified version of the Atkins diet, which we might remember as being very popular in the early 2000s, so much so that they were saying that 1 in 11 people were on the Atkins diet at this time, making it one of the most popular fad diets ever. So years later, we still don't know, is the Atkins diet a good idea or not? And the thing that's cool about long-term nutrition studies and being in Massachusetts is that Massachusetts has really led the way for these studies. So one of the first ones was the Framingham Heart Study led by Thomas Dauber, seen here. And this was started in 1948 with about 5,000 individuals. They then recruited the children and then the grandchildren of the original participants. The other study I'll focus on is the Nurses' Health Study. This was out of the Harvard School of Public Health, led by Frank Spicer and Walter Willett. And the first iteration started in 1976. This is where we'll get our long-term case study from. And then a new iteration started in 1989. And again, you can just look at these sample sizes. Over 100,000 registered nurses were monitored as part of this study. So now we're going to look at the Nurses' Health Study in carbohydrates. And again, when we ask why, here we're looking again at heart disease. We really want to try to prevent heart disease, a leading cause of death and a very big contributor to medical costs. So where are we looking? Here we're looking at nurses from some of the most popular states indicated on this map. And 82,000 women. So a much bigger group than our 150 we talked about earlier. What are we looking at? So things have gotten, again, a little bit more complicated as we've added people. So rather than having our neat two little groups of low carb and low fat, what we have instead is a large group of people that all eat different amounts of carbohydrates, just like sort of a larger version of this room. We're going to monitor them for a really long period of time, and they'll be monitored every 2 years. And how are we going to look at them? So here we can actually look at heart disease diagnosis. This is one of the things that, one of the powerful attributes of long-term studies is that 20 years is a long enough time to expect that heart disease will develop in the population. So patients are going to be sorted into groups based on their carb intake. So if we take 10 groups, we say that the group with the highest carbs will be group 1, the group with the lowest carbs will be group 10, So a group that's eating a low amount of carbs, what would we expect for their other macronutrients? More. Yeah, so again, we're not changing the total number of calories, we're changing the distribution of macronutrients. So these people are going to have higher fat and protein intake. So again, the total calories are similar, they end up being about 1,800. And what they found is that there was no significant increase in heart disease across these groups. And this was a really surprising finding. If you think back to the Atkins news back in the early 2000s, people would say, oh, you'll lose weight, but eating all of that protein and that fat will really increase your heart disease risk. But that's not what they found in this study. So the strengths of this study, we've already touched on a lot of them. So the first is that we're actually able to look at the frequency of disease. We have a long enough time period to measure this. The second is that we have a very large number of subjects. We're looking at almost 100,000 people. And we have a varied population from a number of different states. The weaknesses of this study, other factors may affect results. So this study design is what we call an observational study. So we're looking to see what goes with low carb or low fat diet. There are things that can affect this. So one, we can imagine geography. If everyone in our low carb group is from California and everyone in our low fat group is from Texas, environmental factors could skew our results. Another thing is cigarette smoking. Cigarette smoking is one of the most reproducible causes of heart disease. And so in the analysis, they had to account for the effects of cigarette smoking. The other issue is that these are all women. So as Katie touched on earlier, men and women have different hormones, slightly different metabolisms. So we would wonder if these results would apply to men. And as I said, this is an observational study. So if we compare our two case studies head to head, we see in our short-term study, they were actively changing the fat or carbs in diet, assigning participants to one of two groups, closely monitoring them, measuring short-term changes at about a year. So here again, we're looking at the direct effects of the diet. In our long-term study, here we're artificially just grouping patients by their fat and carb consumption as they report it in surveys. Then monitoring them every 2 years, measuring long-term changes in disease frequency. And so this is where we get something called a correlation. And so if you've ever heard, caffeine is correlated with cancer or something of that nature, these sorts of studies are where those correlations come from. But correlations can be tricky. So we define a correlation by saying that two things tend to move together. So if we look at these two lines, we see that when one thing decreases, the other thing tends to decrease. So this green decrease matched by the blue decrease. And then when one increases, the other one increases. So we would say these two things are correlated. But the question is, are they actually related? So you'd be interested to know that what I'm actually graphing here is the relationship between the divorce rate in Maine and the US consumption of margarine. So I hope this is enough to show. This is actually adapted from a website called Spurious Correlations that has all kinds of these sorts of things. But the point I hope I've made here is that a correlation doesn't imply causation. Clearly, consumption of margarine is not causing divorce in Maine. And so this is one of the things, when you hear about a correlation in the news, you really have to be careful. But this doesn't mean that all correlations are bad. So this correlation I'm showing you now, this is between cigarette smoking and lung cancer in men. This is from the National Cancer Institute. And cigarette smoking, as I mentioned earlier, is one of the strongest indicators of chronic diseases like heart disease, diabetes, and cancer. And so this correlation spurred a lot of research in mice, in cells, looking at the compounds that are in cigarettes and their effects on cells and animals and the body in general. And so we can say that this correlation, it doesn't stand alone. It's supported by other types of scientific evidence. And this is really what we want to look for when we see a correlation. And so the other thing I want to talk about is what we can consider. I told you that I would hopefully make things a little bit clearer so that when you read an article, you can understand what's going on.
So points I think it's especially important to consider. The first is who. What kind of subjects are they looking at? People with diabetes or with heart disease may respond differently to a type of diet than people without chronic disease or younger people, et cetera, et cetera. How? What are they looking at and how do they test it? Are they looking at short-term weight loss? Are they looking at the actual frequency of these things? And then who else? Has anyone shown the same thing? So this gets back to the cigarette example. What other studies support the findings? Science doesn't work in isolation. We're constantly trying to replicate each other's results. And so this is a really important thing to consider. So the take-home points of this section, it's not about the headlines, it's about the studies that they're reporting. Different types of studies fundamentally ask different questions, and that results should be replicable and should fit into a larger scientific context. And with that, does anyone have any questions? We can see. Yes. Yes, so that's a great question. The question is looking at sort of personal monitoring technology like Fitbits and then also using phones to take pictures of food being consumed rather than relying on surveys. I think that's something that we'll definitely see in the future. I've seen at least a few studies where they've used Fitbits to monitor activity rather than just having people report them. I'm not aware of people photographing their food, but I think that would certainly... Right. So yeah, certainly with modern technology, it's a lot easier to sort of figure out where you are and again, adjust the calories you're consuming to your personal parameters. Yeah. Mm-hmm. Right. So basically asking about criticisms of the Tulane study, whether the groups actually limited themselves to 40 grams digestible carbs, and also just sort of criticisms of overall study design. So I'm going to go ahead and talk more about the design just because I think it's more relevant. So as I showed from the graph, the low carb diet is a much more drastic intervention. It's a much bigger change. So some people compared the study to saying, okay, let's have one group eat the same thing they've been eating and then put one group on a diet. Because the low-fat diet is not changing their macronutrients nearly as much as the low-carb diet. And so that was a common criticism of this study. In terms of whether they actually adhered to eating about 40 grams, I can't remember the exact numbers. But yeah, there was a trend over time that they did slightly, their carb intake crept up. But what I showed is the percentage of macronutrients, and even if they didn't reach that 40-gram target, they definitely did drastically restrict their carbs. Yeah. Did you ask a question? Okay. Yeah. Okay, so the question is sort of asking what they were looking at in the second study, if they were looking more at people who were eating large amounts of fat, what they were actually trying to look at. So you're correct in that what they're concerned about is the fat, but the reason why they're concerned about it is because since the participants are eating such low carbs, they're eating more fat.
I actually have a hidden slide for this. So the question is basically talking about, again, variation, but this time not in a person's response, but in what they're replacing the carbs with. And so what we have here is basically a graph that's showing what happens when you replace certain macronutrients with others. And so here at the very top, if you're replacing, actually we'll start here. So we've all heard that trans fat is very, very bad for us. And here, if we're replacing unhydrogenated unsaturated fats with trans fats, if we're replacing trans fats with unhydrogenated unsaturated fats, we see a large decrease in heart disease risk. So this again gets at sort of sources. So it's not enough to say someone's going on a low carb diet. What we want to know really is what they're replacing those carbs with. So we can see based on what you're replacing your different types of fat with, or your carbohydrates, you can see different effects. And so a lot of what this is getting to is that nutrition now is no longer about macronutrients in isolation. What we really want to think about is what we're replacing one macronutrient with. So here, if we're replacing our saturated fat, so 5% of energy, so 5% of our total calories, we see a small decrease in our heart disease risk. But if we're instead replacing monounsaturated fat, so those good fats, that olive oil, avocado, with carbohydrates, we're then increasing our risk. So here, based on what we're replacing, so if we're replacing saturated fat versus unsaturated fat, we're having two different effects. Yes, so we've had enough questions for now. We're now ready for our break. And if you haven't had a chance to participate in our interactive activities, we have the fat content and the sugar content of one of these foods represented visually. And if you'd like to vote on which food you think it is, please go ahead and make a tick mark on the paper. Thank you very much. Sorry, I got really carried away. How do I turn this off?
So welcome back from the break, everyone. My name is Abby Clark, and I'll be going through the third part of our lecture, which is how has society changed food. So our menu for the third course will first decode artificial sweeteners. So we'll talk about what they are and demystify some of the news that's been in the media. Secondly, we'll talk about gluten sensitivity and whether it's real or fake. And lastly, we'll talk about how society has changed food. So how our opinions have been driving changes in the way that food is presented and the food that is available to us. So first of all, to decode artificial sweeteners, what are they? Are there artificial sugars in your food? And are artificial sugars safe? So first, as a definition, artificial sweeteners are 0-calorie sweeteners up to 20,000 times as sweet as sugar, which is an astounding factor. So who can name some artificial sugars that we're all commonly familiar with? Yell them out. Splenda. High fructose corn syrup is technically not an artificial sweetener just because it's not 0-calorie, but it's in the larger category of sugar substitutes. Yeah, equal, exactly. Stevia, ooh, yes, the more modern one. Okay, so yeah, definitely equal. Sweet and Low, Splenda, Truvia, and there's also another one called Sweet One, which is less common but frequently included in food. So what's important is that we're able to identify artificial sweeteners by their chemical names. If you want to identify whether artificial sugars are in your food, you need to be able to look at the ingredient list and be able to identify them. And unfortunately, in the ingredients list, it doesn't say Equal or Splenda or Sweet and Low. So the chemical name of Equal is actually aspartame. For Sweet and Low, it's saccharin. Splenda is sucralose. Sweet One is asulfame potassium. And Truvia is stevia, as you guys already figured out. So the question is, does your food contain sugar or artificial sugars? So something that I like to eat in the morning is oatmeal. And you know that it's really convenient how the oatmeal comes in those individual packets that are already pre-flavored, quick in and out in the morning. Well, a maple and brown sugar oatmeal actually contains two different types of artificial sugar, the asulfame potassium and the sucralose, which you might not be expecting for oatmeal. Another thing that's kind of yummy to have in the morning and you might think is really nutritious is some fruity vegetable juice. So it contains lots of vitamins and minerals to start off your day. But again, if you look at the ingredients list, it does contain sucralose. Lastly, how about some yogurt? Yogurt is so popular right now. We all know that it's healthy with all those probiotics and the calcium. And look how fantastic this is. It doesn't contain aspartame. That's awesome. But if you look at the ingredients list, it does contain two other artificial sweeteners, asulfame potassium and sucralose. So they're trying to get one over on you, but now you know better. So why are artificial sugars calorie-free? As Catherine covered, sugars and starches are broken down into their individual components, and then they're used and traveled in the bloodstream to be used as energy. And this is why they have calories, is because they can be used for energy. However, dietary fiber cannot be broken down into its individual components and used for energy, and therefore it's calorie-free. Similarly, artificial sugars cannot be broken down and used for energy, which is why they are calorie-free. So there have been lots of connections between artificial sugars and weight loss, weight gain, and cancer. But what is the evidence? I'd like to take us through one study for each of these to demonstrate that there is evidence supporting and not supporting each of these. So the first question for this first study that we'll talk about was, does consumption of aspartame aid weight loss? So these scientists were measuring weight loss over 3 years in 163 obese women. And in this study, there was a 19-week active weight loss period. And during this time, these women had intense nutritional counseling weekly. They were given an exercise plan, and their weight was measured weekly. This was followed by a maintenance phase, which lasted from week 19, so when the active weight loss period ended, all the way to 3 years after the beginning of the study. And during this period of time, the women were encouraged to continue exercising, and their weight was again measured, but this time yearly.
So what did the scientists find? They found that artificial sugars may aid in maintaining weight loss. And this is how they depict it here. So they're showing percent change in weight. So it's good if you're on the lower end of the graph, meaning that you've lost more of your body weight. And there are two groups of women here. So there are groups who did not consume aspartame during this active weight loss period. And there are women who did consume aspartame. And in this group, they were counseled on how to include aspartame in their diet on a daily basis. So as we can see in this active weight loss period, women who consumed aspartame and didn't consume aspartame lost the same percentage of body weight. So therefore, we can say that aspartame did not help in losing body weight. However, if we look at this maintenance phase and we skip ahead to 1 year and 3 years, if we look at the women who did not consume aspartame, you can see that by 3 years, they had gained back all of the weight that they had lost during that active weight loss period. However, if we look at the women who did consume aspartame, you can see that they only gained back 5% of the change in body weight that they had lost during that active weight loss period. And this led the scientists to conclude that artificial sugars may aid in maintaining weight loss. But what about artificial sugars and weight gain? Because there's also evidence supporting this. So in this experiment, the scientists were asking, does daily consumption of artificial sweeteners increase the risk of becoming obese? So in this study, they measured weight change over 7 to 8 years, so a longer term study. They looked at men and women ages 25 to 64 years, and the study was conducted in San Antonio, Texas, which, as Mary described, has a high rate of obesity. So in this study, they grouped the participants into 5 different groups based on the number of artificially sweetened beverages that they consumed per week. And so these groups were as such, where some people didn't consume any artificially sweetened beverages, whereas they consumed over 22 artificially sweetened beverages per week. In this study, their conclusion was that artificial sugars may contribute to unintended long-term weight gain. So in this graph, how they're depicting this is the chance of becoming obese. And again, they're dividing the group into 5 different groups based on the number of artificially sweetened beverages that they consume. So what they do is that they set the chance of becoming obese as 1 if you didn't consume any artificially sweetened beverages. And then based on this, what they can look at is, okay, well, for someone who consumed less than 3 artificially sweetened beverages, the risk of becoming obese is slightly higher. This trend continues all the way up for the people who drink more than 22 artificially sweetened beverages per week. And you can see that their chance of becoming obese is increased by 2-fold. So I heard little murmurs when I said 22 or more artificially sweetened beverages. So you might be thinking, how could someone consume that much? Well, it's surprising, but let's say in a day, you consume 2 cups of coffee, and you like to put a packet of Sweet and Low or Equal or Splenda in there. Let's say at lunch, you like to have a diet soda, and then maybe in the afternoon or in the evening, you like to have a diet iced tea. If you were to consume these 4 beverages every single day for a week, that's 28 artificially sweetened beverages per week. So you can see that 22 is not that unreasonable. So lastly, we'll cover artificial sugars and cancer. So in this study, the scientists were asking, does daily consumption of artificial sweeteners increase the risk of getting cancer? So in this study, they were looking at the development of cancer over 18 months in male and female rats. And what they did was they gave the rats food with or without saccharin. So they found that artificial sugars may contribute to cancer in rats. And in particular, what they found is that it in particular pertains to bladder cancer. And so again, we have 2 groups. So we have rats who were given food without the saccharin, and then rats that were given food with the saccharin. So what they found was for females who didn't consume any saccharin, none of them developed bladder cancer. Whereas the females who did consume saccharin ended up developing bladder cancer. If we look at males, the story is much different. So for the males who didn't consume saccharin, approximately 3% developed bladder cancer. But astoundingly, 30% of the rats who consumed saccharin during this period of time ended up developing the cancer. So you might be really worried about this. You're saying, oh, well, I really like to put sweet and low in my coffee. Does this mean that I'm going to get bladder cancer? So first of all, you should realize that the amount of saccharin that these rats were exposed to was up to 1,000 times more than someone might consume in any day. So it's a crazy amount. There's no way that one person could consume that much sweet and low in one day. There have been many subsequent studies that have demonstrated that these results are not relevant to humans. And so in fact, there was a cancer risk notation that came along with sweet and low for a while, but it has been subsequently removed because evidence has demonstrated that the studies and the results are not relevant to humans. So as sort of a thinking exercise, I'd like to take us through a typical day of artificial sweeteners, harkening back to the idea that we might be consuming more artificial sweeteners than we might realize. So in a typical breakfast, again, you might start off with coffee. And you know those big Dunkin' Donuts coffee, I bet people probably put 2 packets of some sort of artificial sweetener in there. So let's say that that's maybe 4 artificial sweeteners by the time you have lunch. Oatmeal or some fruit vegetable juice. Again, both of those contain the artificial sweetener. Let's move on to lunch. Maybe you like to have a couple diet sodas, maybe one with lunch, one in the afternoon, pep you up a little bit. Well, it might surprise you to know that each diet soda contains the equivalent of 6 packets of Equal, which is the most common artificial sweetener used in diet soda. And let's say in the afternoon you like to have the light yogurt. Well, we already know that this also contains those artificial sweeteners. Let's say for dinner you want to be really healthy and have a salad. So you say, great, I'm going to have some salad with some fat-free salad dressing. Well, it should be noted that the majority of fat-free salad dressings contain artificial sweeteners to make up for the fact that they don't have fat in them. And let's say for dessert, one of my favorite desserts is rice pudding. And so let's say you're trying to be really healthy and you make it with some Splenda to reduce the calorie content. It's likely about 2 servings of your artificial sweetener in that dessert. And so the point that I would like to make with this part of the lecture and really drive home is that these artificial sugars and these artificial sugar packets that we all put in our food every day, while they might be safe, as determined by recent experimental studies, it doesn't necessarily mean that they're healthy. And so again, we need to think about everything in moderation. And so with that, I'd like to take any questions that might have come up. Sure. Sure. So the question is, in the study with the number of artificially sweetened beverages, were they controlling for the amount of regular sugar beverages versus artificially sweetened beverages? And so they grouped the participants who were the all sugar beverages into that none category. So they were the no artificially sweetened beverages. Now I understand what you're saying. So the question is, were they consuming the same amount of calories? And there was a slight difference in the amount of calories, but not necessarily specifically significant that would result in the finding of the test. Sure. So the question is, are these artificial sweeteners perhaps chemically in the body lead to some sort of change that might contribute to a cancer risk? And honestly, I'm not quite sure. So the types of studies we need to do in humans, as Mary was getting at, were correlation studies. So it wouldn't necessarily be ethical to take people and give them, 25 months to see if they develop cancer. The correlation studies haven't shown a link between artificial sugar and cancer. In terms of evaluating it on a more level, maybe a model. I'm not sure what those studies show.
trying to maintain that in the study where it was the weight loss maintenance and either consuming or not consuming the artificial sugars, where they replaced the regular sugar with the artificial sugar. And so in fact, they were replacing the artificial sugar or the regular sugar for the artificial sugar. So maybe your question is, well, obviously you're reducing their calorie content, but in fact, that's not necessarily the case. As you can see with the subsequent study where it was the unintended weight loss, that even though you might be you think that you're consuming fewer calories because perhaps you're taking out that Coke that might have, or regular soda that might have 150 or 200 calories and it doesn't have any calories, you might be overcompensating for that by having a little bit more for dinner. So it's not necessarily a one-for-one kind of replacement situation, which is what a lot of these studies have shown. So with that, I think we'll move on to the next portion, but please save your questions and come ask me after the presentation. And we will have another question break afterwards. So back to our menu for the third course, we'll next transition to gluten sensitivity. So first, we'll define what gluten is. We'll identify what food contains gluten. We'll talk about what celiac disease is. Is gluten sensitivity real, which has been a really hot topic in the news lately? And then we'll talk about whether it's safe and healthy to be gluten-free. So gluten is a protein. It's composed of two parts, gliadin and glutenin, and it acts like a glue in your food, keeping everything together, which is why it's a lot of baked goods. So gluten can be found in all wheat-containing products. So this includes breads, cookies, bagels, pasta, wheat cereals, as well as barley that's used in making beer or rye. Importantly, however, it is not found in oats, potatoes, beans, corn, or rice. So celiac disease is an autoimmune disorder that is an allergy to gluten. So again, all of the gluten contained in these foods. It affects about 1 in 100 people, depending on what area of the world you're in. Symptoms can include fatigue, gastrointestinal pain, and malnutrition. And importantly, it can lead to long-term damage of the small intestine, which is why it's important to diagnose celiac disease. The treatment for the disease is to abstain from eating gluten, and it's been demonstrated that taking gluten out of the diet permanently can actually restore and take away some of that damage to the small intestine. And again, it removes all of the symptoms that I discussed before. So gluten sensitivity is really similar to celiac disease, and then it has similar symptoms when someone eats food containing gluten. But very importantly, it doesn't lead to damage to the small intestine. So again, it's all about these symptoms that you're experiencing, but you don't have this damage to the small intestine, so the symptoms might be a little bit different. So the treatment, again, is just to stay away from gluten and remove the gluten from the diet, and this should remove the symptoms that you've been experiencing. So let's look at the science. What does the science say about gluten sensitivity? Much of this work has been pioneered by Peter Gibson in Australia. And in a 2011 study, they measured gastrointestinal symptoms in patients whose symptoms have previously improved on a gluten-free diet. So the study was designed as follows. So there was a 2-week period before the start of the study where all participants were put on a completely gluten-free diet. And this was designed to make everybody's baseline symptoms as close to zero as possible, so to have people feeling as great as they possibly could. In the next phase of the study, it was broken into 2 groups. So it's important to note that both groups maintained a background gluten-free diet. The plus gluten group ate a gluten-containing supplement each day, and the minus gluten group ate a gluten-free supplement each day. And these were both of equal calories and about equal taste. During this 6-week period where they were either eating the gluten-containing supplement or the gluten-free supplement, participants maintained a log of gluten-free foods eaten as well as their symptoms. After the study, the scientists then analyzed the data
based on the symptom log that the participants had created. So they found that a gluten-free diet can reduce overall symptoms. So what they're depicting here is the extent of symptoms, and again, this was based on the symptom log that had been kept by all of the participants. So in red, we have the patients or the participants who had the gluten supplement, and in blue, we have the participants who have a gluten-free supplement. And what you can see here is that the people with the gluten-containing supplement had far more overall gastrointestinal symptoms than those who had the gluten-free supplement. So this supported the notion that a gluten-free diet may reduce overall symptoms in these participants, in these patients. But it's important to note that gluten-free does not always mean healthier. So I'd like to play a little game, and it's which has more. So let's say you're in a grocery store and you want to have a nice spaghetti dinner, and you've recently been listening to the news and you've heard that gluten-free is better for you. So you're thinking, okay, I want to have pasta, but maybe I'll buy the gluten-free one and it'll be better for me. So we'll look at Barilla spaghetti. So again, for the same serving size, both 2 ounces, both have 200 calories. Which do you think has more fat, the gluten-containing or the gluten-free? Really? Tricked you there. Gluten-containing actually has more fat. Okay, what about total carbs? Gluten-free, gluten-containing? Yes, gluten-free, good. So then what about fiber? Gluten-free or gluten-containing? Gluten-containing, exactly. There's far more fiber in wheat than there is in corn or rice. So a couple other things to importantly point out, that yes, the gluten-containing pasta has more sodium and slightly more sugar, but it also has more protein, 7 grams compared to 4. And as Catherine pointed out before, protein is really important in our diet. So we can also look at some whole grain bread, so we can compare gluten-free versus gluten-containing. And as you can see, the gluten-free bread is overall far worse for you than the gluten-containing bread if we think about the number of calories, the amount of fat, sodium, total carbs, as well as sugar. And lastly, to really illustrate this, we can think about pie crust. Maybe Thanksgiving is coming up and you're thinking, oh, I really love that pumpkin and that apple pie. Maybe I'll make it a little bit healthier by making it with a gluten-free crust. Well, you wouldn't be doing yourself much good because it contains 5.5 grams more fat than the gluten-containing version. So maybe just eat a smaller piece. So the point that we want to make with this section is that gluten sensitivity is likely a real condition for some people, and that it shouldn't just be thrown out as a non-condition and that it doesn't exist. And the other thing to remember is that gluten-free does not mean healthier. There are many other things to take into consideration that's been outlined in other parts of this talk about getting a good balance of your macronutrients. And so before I end the talk with a quick discussion about how society has changed foods, I'd like to take a few more questions. Yes. Right. So the question is it likely that gluten sensitivity always existed? Because all of a sudden it seems like it's everywhere. So part of this is likely due to the fact that it's more recognizable now. So people are paying attention to these gastrointestinal symptoms and trying to figure out where they come from. So a certain part of it is definitely recognition. But another big part is the fact that we're eating more things containing wheat and gluten. And so perhaps that's exacerbating all of these symptoms. Sure. Right, so the comment that she's making is that perhaps it's not that we're eating more, but the things that we're eating have a higher gluten-containing content. And that might, in fact, definitely be true. Sure. Mm-hmm. Yep. So
the question is whether stevia, which is really a natural artificial sweetener, is considered any more healthy than the other artificial sweeteners like Sweet and Low or Equal. In terms of the healthiness, I'm not sure. I'm not sure whether they have been used in these studies for long-term weight gain or perhaps long-term weight loss. So I think that has yet to be seen. And since we don't see a cancer risk between the other sort of more historical artificial sweeteners and cancer, I wouldn't necessarily expect it with the natural sweetener either. Okay, so with that, we'll end the talk. And please, if you have any more questions, feel free to come up to me afterwards. So how has society changed food? So you may have heard in the news lately that there have been some proposed changes to the nutritional food labels. So this is the old nutritional fast food label that we're all familiar with, and this is the new nutritional label. And some obvious things that might be different are that, first of all, the servings are larger. So it's clear that there are 8 servings per container in this particular food. In addition, the calorie type is larger, and added sugars is a new component of the label. So not only are we understanding how many sugars are in the food, but how many sugars, how much grams of sugar the manufacturer has added. And so really this has been driven by our intense interest in nutrition and understanding what's in our food. Further, we can think about gluten-free menus, because again, this has been driven by society's interest in reducing gluten in their diets if you have gluten sensitivity, or even the increased awareness of celiac disease. And so some restaurants that are right in the Boston area with gluten-free menus, the Franklin Cafe for 2 Cheese, Citizen Public House, Legal Seafoods, and even Olive Garden, the pasta house of America. So I think this is really great because it enables more food choices for people who have celiac disease and gluten sensitivity, and it opens up the possibilities for going out and dining. So some take-home points from this third course. Artificial sugars may be consumed in limited amounts safely, but it doesn't necessarily mean that they're healthy. Gluten sensitivity may, in fact, affect people without celiac disease. However, the explanation remains unclear. Science has provided this evidence for changing our food, but importantly, society has driven these changes. And lastly, we'd like to end the talk with some take-home points from each of our 3 parts. So as Catherine discussed, carbs, proteins, and fats are fuel to provide our bodies with energy. And remember that excess calories are converted to fats. As Mary discussed, it's not about the headline, it's about the study. And so it's important to critically evaluate these studies in order to get away from the hype and figure out what the truth is. And lastly, I think it's about making informed decisions. So we're aware more than ever of what's contained in our food and what we should be eating and what we shouldn't be eating. And we do have the power to change what's in our food and what we purchase on a daily basis. So with that, we'd like to say thank you for coming. It's been great having you as an audience. And really quickly, I'd just like to point out that we can talk about our winners for the little game that we have you guys play. So, just as a reminder, is to identify which drink contained this much sugar. So in fact, this is 50 grams of sugar weighed out. This is the amount of sugar in a 16-ounce glass of orange juice. And importantly, what I'd like to show is that the smoothie and the Coke or the soda contain the same sugar as the orange juice. So basically, this is a trick question in that you were right no matter what you picked. You like that one? So then the other one was to identify this guy. So how much fat is in this one? This is actually 33 grams of fat. And in fact, this belongs to the grilled cheese sandwich, which was the astounding winner in this poll. There are 13 grams of fat in a hot dog, and there's approximately 18 grams of fat in a slice of meatloaf. And the goal of this exercise was to be able to visualize the grams of fat and sugar that are in the food that we consume. So you might look at the nutritional label and see on a bottle of a fruit smoothie that it contains 50 grams of sugar. And so you might be thinking, well, that sounds like a lot, but maybe it's not really. I think the next time you go to have a smoothie and you think about this much sugar, you might rethink that decision. So with that, we'd like to say thank you for your attention, and please come up with any more questions.
Thank you for tuning in for tonight's Science in the News lecture series. We'll be back next Wednesday at 7 p.m. with a talk on fracking. If you have any more questions, please feel free to fill out the form and we will have our speakers respond to you through email. Thank you again for joining us.